Hello. We're starting our section on muscle, and the focus is specifically skeletal muscle in chapter 9. And that's what we see here as the image here in the background. It's just a very close magnification on skeletal muscle tissue. So to begin, we want to point out that there are different types of muscle tissue in the body. Skeletal muscle is the focus for this chapter and unit. One type of muscle, cardiac muscle, is unique to only one organ in the body, the heart, nowhere else. And this is, of course, the muscle responsible for contraction of the heart and our blood pressure, so providing the power for our circulation. And we'll study this in a later section. A second type, there's smooth muscle. And smooth muscles found in many structures. Many of them are hollow tubes, such as the uterus, the stomach, and you can see here on this diagram, blood vessels, arteries, and veins. And in the wall of that structure, there's a layer of smooth muscle. The erector pili muscle in the skin is another example. But the focus for now, of course, is on skeletal muscle. And skeletal muscle, by and large, connects to tendons, and in turn connects onto our skeleton, onto our bones. So in our body, there's about over 700 unique skeletal muscles, collectively making up the muscular system. And again, this will be the focus of this section, is skeletal muscle tissue, and that'll lead us right into the study of the muscular system and the large muscle organs. So now focusing on skeletal muscle and the muscular system in particular, multiple functions for this system and thus this type of tissue. First one is movement of our body. So our ability to move our flexible joints, that comes from the power and force produced by skeletal muscle. We're able to maintain our upright posture. So humans, we're able to stand up on 2 feet and maintain our upright, erect posture. So muscles on our legs, our torsos, and coordinating all of those muscles to maintain our posture. Areas that lack bones specifically are a great example of where skeletal muscle does help to protect and support structures. And a great example would be the abdomen. So where we have all the various abdominal muscles between the rib cage and the pelvis area that's lacking bones, that's supported by layers of skeletal muscles. So that's one good example. An orifice is an open canal. So you want to think of your mouth, for example, the nostrils, the urethra. And these openings, whether it be an entrance or exit point to the body, they're controlled by the contraction of skeletal muscle. And the final point here, during contraction, our skeletal muscles use a tremendous amount of energy in the form of ATP, the typical energy source for our cells function. And by expending so much energy, by breaking down ATP, this is a way to give off heat. It gives off energy, and we interpret that as our body temperature. So our contracting, worsening muscles is the source of our warm temperature. And just a simple example, you exercise your muscles, you heat up, and then we use our skin, sweat glands to perspire and cool off. So now moving through this section, we're going to start with the cytology of skeletal muscle tissue. So looking at its microscopic structure. And a helpful figure that we'll go to is this figure 9.3. And we're now just describing structures of a skeletal muscle cell. And you'll note that many times we're going to be using these prefixes, both sarco and myo. And both of these pertain to muscle. So as we start out, we could begin here with what's called the sarcolemma. every cell in the body has a plasma lemma or a plasma membrane, a cell membrane as well. And we see just this specific term used for the cell membrane of a skeletal muscle cell is the sarcolemma. There's the sarcoplasm, simply just meaning the interior space of a muscle cell. All of the area inside of the sarcolemma is the sarcoplasm. And when it comes to skeletal muscle in particular, they are these long cylindrical cells. So we can't interchange the word fiber or cell when it comes to muscle tissue. So as I mentioned, these long cylindrical cells, that's the shape of a mature skeletal muscle fiber. And it also is a multinucleated cell. Along the length of that long cylinder, there are multiple nuclei in a mature skeletal muscle fiber. And how this develops is due to the fact that we have these precursor cells called myoblasts. So myo meaning muscle, and blast, of course, the younger, newly developed cell. These precursor myoblasts each contain a single nucleus. And as multiple myoblasts then bond together, they start to develop into a larger, now multinucleated cell. And this process continues several times over to make the mature, elongated, multinucleated skeletal muscle fiber. And here's a much better figure. You notice this picture 9.3. Each myoblast has its own single nucleus. They're bonding together to make a multinucleated cell. The cell membrane, known as the sarcolemma, and all of the interior space within the sarcoplasm. So now we notice within the sarcoplasm, we start to see now these bundles known as myofibrils. One skeletal muscle fiber is filled with, you notice here, just dozens and dozens of myofibrils. So going into a smaller scale, the myofibrils themselves are made of bundles of multiple myofilaments. And they come in the two varieties. You notice there's a thick myofilament and a thin myofilament. The thin myofilament is composed of three components. There's three parts to it. The principal structure of the thin filament, however, is a protein known as actin. Whereas now thick filaments, these are now just large bundles of a protein called myosin. And together, these two myofilaments are arranged in a precise manner to make the larger myofibril structure. But now we can notice that the entire myofibril itself is wrapped in now these other features, these other organelles. And that's where we'll touch on the sarcoplasmic reticulum, the terminal cisterna. And let's continue forward. So wrapped directly around the myofibrils, there's now the multiple sarcoplasmic reticula, commonly abbreviated to SR. And this is truly equivalent in structure to the endoplasmic reticulum that are found in a typical cell, but again, using that prefix for muscle. The sarcoplasmic reticulum wrapping around the structure of the myofibril. And their key function is they store, and more importantly, they release calcium ions. And this calcium release is a vital step in order for the muscle cells to contract, to shorten, and to produce that force for motion. Bordering on the edges of a single SR are now the terminal cisterna. And we'll go back, of course, to the textbook figure. But right here is a sketch now of a sarcoplasmic reticulum. And the expanded edges on each side are known as the terminal cisternae. Just these expanded edges of the SR. And running right along the terminal cisternae are a network of canals, a network of tubes, known as transverse tubules. So if we add to our sketch, we'll put a transverse tubule boarding right along each terminal cisternae, and then we'll draw the next sarcoplasmic reticulum. And that leads us to this final trait here for this slide, a triad with 3 parts, of course, 3 components. A triad is now the union of 2 adjacent terminal cisternae and the transverse tubule that's running between them. So we'll highlight that right here. So that union of 3 parts, 2 terminal cisternae, a single transverse tubule, that union with 3 parts is known as a triad. And we'll go to the textbook figure once more. So we can start with the myofibril, composed of thin and thick myofilaments, all bundled in the calcium storing sarcoplasmic reticulum with this expanded edges known as the terminal cisternae. Running between them here, highlighted in yellow, are the whole network of transverse tubules. And you can see that unit there of the triad. So going back to the description of T-tubules, we can see that a T-tubule, it truly is a connection between the sarcolemma, the cell membrane of a muscle fiber. They start at the surface, and now those transverse tubules penetrate all the way deep in, and they run right along the terminal cisterna. And that's truly their purpose. They're an ion channel that's connecting those 2 areas. An ion channel between the sarcolemma and the terminal cisterna. And just a closer look at that here. And even closer in. So starting at the sarcolemma, which leads directly into a transverse tubule, and bordering right against that terminal cisterna. So now we're going to take a look just at the larger scale view of what's going on here for muscle contraction. In order for a skeletal muscle fiber to begin its contraction, it needs to receive a signal from the nervous system. And this comes via a neuron known as a motor neuron. This is a cell of the nervous system that's signaling from the brain or spinal cord, sending this signal down to the skeletal muscle, ultimately to stimulate contraction. So in this first step here, we're looking right in this area known as the neuromuscular junction. It's where the nerve cell and the muscle cell communicate to one another. The nerve cell is releasing a chemical, this neurotransmitter. And for muscle contraction, the specific neurotransmitter produced is known as acetylcholine. And we'll use this abbreviation here, just ACH, standing for acetylcholine. It's released out of the nerve cell, and it binds to a receptor that's found on the sarcolemma, on the surface of the skeletal muscle fiber. Ultimately, this opens up ion channels on the sarcolemma. So now ions are able to penetrate into the muscle cell, and this leads to an electrical current. And ultimately, this electrical current is what stimulates the contraction function of the skeletal muscle fiber. And the details here, of course, that's now getting into the physiology. So we won't go any further than this. Just the production of the neurotransmitter ACH, interacting with the sarcolemma, and triggering the process of contraction, the shortening of the skeletal muscle cell. However, this is not a realistic portrayal to see one motor neuron communicating to just one muscle fiber. Instead, we have what's known as a motor unit. And a motor unit is described now as the total number of skeletal muscle fibers that are controlled by an individual motor neuron. And there could be variation in the size of a motor unit. The smallest possible motor unit would only have 3 cells, meaning that one cell is the motor neuron, and now the 2 skeletal muscle fibers that it's controlling. So here's a very small size motor unit. A single motor neuron is going to signal and release ACH on these 2 different skeletal muscle fibers and stimulating these 2 cells to contract simultaneously, to work as a group, as a unit. Whereas the largest possible size motor unit would be 1 motor neuron that's controlling up to 2,000 skeletal muscle fibers simultaneously. The benefit here being a very small motor unit, just only controlling a few number of cells, could lead to very, very fine, precise motor functions. And think about the functions of your fingers. Like if you're typing or playing a musical instrument, you can have that very precise, controlled, subtle motion. Whereas a very large motor unit, contracting up to a few thousand cells simultaneously, large motor units could generate a lot of power, a lot of force. So now moving on to the structure of the skeletal muscle fibers, we'll go a little deeper in. And we'll discuss what's known now as a sarcomere. So just once again, seeing that prefix, sarco. This is now the unit of physiology, the unit of contraction. So once a skeletal muscle cell is contracting, it's truly the changes that go on within the sarcomere that actually lead to that shortening of the entire cell. This is related now to the previous structures we were just looking at, the myofibrils, themselves made of the thick and thin myofilaments. So myofibrils are now segmented into multiple sarcomeres. In fact, it takes about a few thousand, several thousand sarcomeres connected end to end to make just a single myofibril. So at this point, we're describing something that is extremely tiny, these sarcomere structures. But you can see the concept. If each of these sarcomeres has the ability to contract, and their contraction only shortens just by a few tenths of a micrometer, but with each sarcomere contracting, and this happens over the length of multiple thousand sarcomeres, that'll make the entire myofibril shorter. So a simple analogy is, if you think of a chain is made of a series of links. So these links are connected together in series, just meaning end to end. So multiple links together make this entire chain. And if we can make each link shorter, then you can see how the entire chain would be shorter. Or another simple idea is, think of a train. The entire train is formed by the linkage of several cars. So if you had shorter cars, you'd have a shorter train. So that's what we'll move into next now, is the microanatomy of an individual sarcomere. And it's labeled with all these different letters here. You notice Z, I, M, H, A. We'll go through the various parts. And again, keep in mind the sarcomere is just one tiny little segment of the much larger, much longer myofibril. So we'll begin here. This is a good figure to start with. We'll begin with what's called, it could be called an I band or a light band. Those could be interchanged. You notice you spell the word light with the letter I, to help you remember that. The I band area, first of all, the I band actually is shared between neighboring adjacent sarcomeres. So it actually overlaps neighboring sarcomeres. And the I band, this only contains thin filament. There's never thick filament in this region. Whereas now the dark band, the A band, this is the area that contains all of the thick filament located here. So you notice the labeling here, the A band, and the purple lines here are representing the thick filament. The border of the sarcomere, right at the edge of the sarcomere, and it appears here as kind of this zigzag line, that is known as either a Z line or Z disc. Again, all of these terms can be interchanged here. This is now the border of the sarcomere. This is where one sarcomere is connected to its neighbor, is at the Z disc. And another trait is the thin filament. The thin filaments are now connected to the Z lines. And that's represented here as the red line, the thin filament. And the thin filament is always connected on to the Z lines. So these first few parts here, I band, A band, and the Z disc. Just a closer look. We stated that the thin filament is made of three principal parts. The actin is the principal structure. The major structure here is represented by these pink circles. However, wrapped around the actin, there's a second structure known as the tropomyosin, as well as the troponin. And we'll take a look at each of those. The thick filaments, again, the full span of the thick filament, that's located here in the A band that we covered earlier. Overlapping adjacent sarcomeres, that's the area of the I band. That's where we only have thin filament present. The Z disc, as we just pointed out, that's the boundary of one sarcomere is from Z to Z. And the newer terms we're adding in now, the M line. The M line is the center of the sarcomere. And the M line is the connection point for all of the thick filament. So the myosin thick filament is all centrally connected to that M line. And then lastly, the H zone. The H zone is a smaller area within the A band. And what's unique about the H zone, this is the area of the sarcomere that only contains thick filament. So represented here by the purple lines branching off of the M line. The H zone only contains thick filament, myosin, whereas the I band only contains the thin filament, the actin filament. So it's with all of these components here, There's the border of the sarcomere, the Z disc, the area that contains just thin filament, the I bands, the center of the sarcomere, the connection point for the thick filaments, the M line. All of the thick filament is bound within the region of the A band. Within the A band, the area that only contains thick filament is the H zone. So it's these 5 areas here. Taking a closer look at each of the myofilaments, there's the thin myofilament. And we, again, described how there's 3 components, and here they are. The majority is composed of actin. Wrapped around the actin is the tropomyosin. And in turn, connected to that is troponin. We'll go back to the illustration to see this, but the actin itself is now just physically blocked by the position of the tropomyosin molecule. And in fact, it's covering the active sites that are on actin. These binding sites are just physically blocked. As long as these active sites are blocked, the sarcomere and thus the muscle cannot contract. And that's the job. Tropomyosin is preventing contraction. Whereas this third protein, troponin, it's holding the tropomyosin in place. So it's aiding in the position of the tropomyosin, which of course is blocking those active sites. So the way to remove each of these is with calcium. So calcium directly interacts with the troponin. So this no longer holds the tropomyosin in place. The tropomyosin can now be released to expose the active sites on actin. So calcium is the source of removing both of these 2 proteins here. So we got to think about where it's the source of this calcium. And that is the storage site of the sarcoplasmic reticulum, which is directly wrapped right around the entire myofibril. And again, with the release of calcium ions out of the SR, calcium directly interacts with the troponin to remove tropomyosin. And now we could expose the actin filament. And we'll see that here again. So a thin myofilament, principally made of the actin, in a resting, relaxed muscle. Calcium ions are stored in the sarcoplasmic reticulum. So now we have the tropomyosin just physically bound to the active sites, blocking that location. And tropomyosin, again, maintaining that position. As we just described from the SR, there's the release of calcium ions that directly interact with the troponin. Leads to the disassociation of troponin and tropomyosin to expose these active sites. And this is now where the thick myofilament comes into play. The thick myofilament, made of large bundles of myosin proteins. The myosin, as we just discussed, is connected centrally in the sarcomere to the M line. And we'll sketch that out here. The myosin itself, the side connected to the M line, is known as the myosin tails. And the opposite edge is known as a myosin head, sticking out on either side. And once the actin has been exposed, the myosin head has the ability to attach on to actin. Again, that can only occur after the release of calcium. So at this point, we can now have an attachment, a cross bridge between the myosin and the actin. The thick myofilament and the thin myofilament are now connected to one another. And we can see that here in the illustration. So with all of these multiple myosin heads, now with the potential to attach onto actin, the next step is now, we notice here on this figure, the myosin head has this ability to move at the hinge. So once connected, the myosin, you can imagine on this side of the sarcomere, attaches on to the actin and can pull that actin towards the center of the sarcomere. And this happens on both sides. So an isolated muscle cell, even an isolated sarcomere in theory, would pull both sides of the thin filaments closer to the M line, closer to the center of the sarcomere. And if we were to do that, that would reduce the overall length of the sarcomere. That would lead to the contraction of the sarcomere. And that's truly the theory of contraction. So we're just going to touch on the overall outcome. We won't get into the detailed steps at this point. So when a skeletal muscle is contracting, think of your biceps on your brachium as you're contracting to flex your elbow. Well, occurring at the cellular level, occurring within the sarcomeres. The I-band segment, that is reduced in length. The Z lines are being brought closer together. The length of the sarcomere is being reduced. The sarcomere is shortening. However, the thin and thick myofilaments stay the same length. So these elements are not contracting. It's just truly their position relative to one another is changing. Much more of the thin myofilament is getting pulled into the A band. More of the thin myofilament is getting pulled closer to the M line. And a description here, and we'll see this visually. So again, with the storage of calcium in the sarcoplasmic reticulum, that calcium's released out and interacts, of course, with the troponin, releasing tropomyosin. Myosin heads attach on to actin. Actin is always connected on to the Z lines. So we now begin by pulling the actin closer to the center of the sarcomere. And again, that connected actin always bound to the Z lines. When we're pulling the actin closer to the center of the sarcomere, we're also pulling those connected Z lines as well. And reducing the length between one Z line to the next Z line, reducing the length of the sarcomere, is contracting the sarcomere. And again, you want to extrapolate this to the fact that multiple thousands of sarcomeres all linked together make one myofibril. And again, if each sarcomere is contracting, the entire myofibril will shorten. And a great illustration here. So a relaxed sarcomere, a resting muscle, and we see this distance between Z line to Z line, the length of a sarcomere. And as we just described, myosin heads pulling actin towards the center of the sarcomere, dragging those connected Z lines, and we see that reduced length of the sarcomere. However, the length of the myofilaments, thin and thick, remains the same. The eye band area is reduced in size. And you also notice here, the H zone is also reduced in size as well. More actin is moving into the A band, reducing the size of the H zone. And here's just a nice animation kind of putting these parts together. So we'll go through this. You notice the color coding here on the side. As our sarcomeres are contracting, again, truly what's happening is the myosin is attaching on to the actin, pulling it towards the M line. And we notice here in red, here's our myosin molecule. This would be the myosin tail. The M line is off to this side of the screen. We have the myosin head, which you notice has this ability to move. That's connecting on to actin, represented here. And you notice by pulling and releasing and pulling, we're now just pulling that actin closer to the M line. Wrapped around the actin are now the troponin and tropomyosin. And to release them, this is the need for calcium. So we can imagine that our sarcoplasmic reticulum would be somewhere over here. And we're releasing this yellow little circle representing calcium. Calcium first directly interacts with the troponin. That now adjusts the position of tropomyosin. And it's only at that point the myosin head attaches onto actin. The final player that we are concerned with is now the energy source here. You notice in green ATP. So produced in large numbers in the cell, the mitochondria specifically. And every time the myosin head moves one direction and back, that's using the energy source of ATP for that activity. And you have to now extrapolate that over just millions of myosin heads using so much ATP, giving off so much energy, and adding to the production of heat from our muscles. Moving forward now, we're going to start to take a look at the larger structure of a muscle organ. So moving beyond just the cytology, moving up to an organ. And there's large amounts of connective tissue. You want to think of collagen, elastic fibers, used to bound just multiple skeletal muscle cells together to create muscle organs. So starting at the superficial surface, the entire muscle organ is wrapped in a layer of connective tissue. And you notice the prefix here, epi. So at the surface of the muscle, the epimysium, just bundling, wrapping all of the muscle cells, stabilizing that entire muscle organ. So such as an entire biceps muscle wrapped in epimysium. Taking a cross section through, and we will use this figure 9.1 to represent this. Taking a slice through the muscle, there's now these smaller bundles known as fascicles, just meaning a bundle of cells of some type. And each individual fascicle is now wrapped in a layer of connective tissue. And you notice the prefix here, perimysium. So bordering each fascicle. And then going to even smaller scale, if you break apart a fascicle to individual cells, each skeletal muscle, of course, has its sarcolemma, and the sarcolemma is wrapped in this deepest layer of connective tissue known as endomysium, the internal deep layer, endo. So it's ultimately these three layers of connective tissue are used to bundle just thousands and thousands of cells together to build a complete muscle organ. And along with the muscle tissue and connective tissue, There's also the blood vessels, the nerve supply, and these structures as well have to penetrate through the layers of connective tissue so that the nerves could signal to their motor units, and of course the muscles can receive their nutrient supply and energy supply. So to see that here, we'll start at the large scale. So the entire muscle organ wrapped in that external layer of the epimysium. You could break it apart to the multiple fascicles. That's what we see here. And a fascicle wrapped in perimysium. So bundling this group of cells together. The fascicle could be broken apart into individual skeletal muscle fibers, individual cells, and one muscle fiber, of course, wrapped around the sarcolemma is that deepest layer of tissue, the endomysium. Now we can just work through this here. So at the big scale, looking at the entire muscle, wrapped in the epimysium, partitioned into smaller fascicles. You notice the nerves, the blood vessels as well. So now looking at an individual fascicle wrapped in perimysium. Again, it could be broken down into an individual skeletal muscle fiber. And that's where we are now, looking at one cell now wrapped in the endomysium. And leading back to about where we started, one cell containing multiple myofibrils. One single myofibril is segmented from Z line to Z line into multiple sarcomeres. And that's what we see here. Breaking down a myofibril, and from Z to Z, an individual sarcomere. And the sarcomere itself, we cover the different portions, Z lines, I bands, M lines, A bands, and H zone. Now, ultimately, it's the muscle tissue that contracts, but now we need the tendon structures, or the aponeuroses, to have a connection on to some other structure. The muscle's contracting, but we need to be connected to something to pull on. And that's where the tendons and aponeuroses now come into play. So the general description for a tendon, it's dense, regular connective tissue that connects skeletal muscle, typically to bone. This is the usual case. So pulling on those various bone structures that we learned, tubercles and tuberosities and trochanters. However, in the terms of facial expression, like if you want to smile, any type of facial expression, that's muscles pulling right on your skin. And in other cases as well, pulling directly from other muscles. And we'll see some of these examples in lab. An aponeurosis now, this is truly just a distinct shape of tendon, and that's what you see here on this little clay model figure here. It's just simply a very broad, flat tendinous sheath, such as the one that we have here on the superior portion of the cranium. This would be known as the gallia aponeurotica. And we have aponeuroses over the abdominal region, the anterior abdominal region. There's one on the lumbar dorsal region. Whereas this figure here, the typical tendon are just these thin cords. Aponeuroses is a broad sheet, but they have that similar function. This next topic is now looking at the shape of muscle tissue structures. And we see these various shapes all the way at the surface of the muscle organ. So depending on how the fascicles, the bundles of muscle cells, their orientation and connection to the tendon, we can see these 6 principal shapes, these 6 principal appearances for muscle organs. So we have the description here, and then we'll skip to a figure that shows these. So to begin, what's known as parallel muscle, the fascicles of muscle cells, they are arranged parallel to the length of the entire muscle organ. So a simple sketch, you have a tendon on one side, another tendon, and the skeletal muscle cells. I'll switch colors to make it kind of distinct. Skeletal muscle cells run parallel to the length of this entire muscle organ. So kind of like so. Convergent, described as having a broad base on one side, and it tapers down to a smaller area on the other side. So roughly a triangular shape. There's the wider base, tapering or converging down to a smaller area. And circular, just like the name describes. There's now a circular ring of skeletal muscle around some orifice, around some opening. So your lips, your mouth are a great example with that circular orientation. And let's go right to the textbook figure here. So what I was trying to draw for a parallel muscle shape, a convergent shape again starts with a broader base, tapering down to a smaller area. This cross section will become important in just a moment. And a circular ring, you notice that its function is to close off and constrict some orifice. So now these latter 3 shapes here, the pennant shapes, uni, of course, meaning one, the muscle fascicles connect to the tendon, running at an angle to one side, bipennate, branching at 2 separate angles on each side of the tendon. And multipennate now, if you look at this first line of description, multipennate muscle appears at the surface just like convergent. There's a broader base and it tapers down to a smaller area. The distinction now is in multipennate muscle, the tendon is not simply located on each end of the muscle. The tendon actually branches right through the center of the muscle itself. So if we go back to the illustration for convergent muscle, I mentioned a cross section slicing through the center mass of the muscle. There's no present tendon. And we'll skip forward to multipennate. A broad base tapers down to a smaller area, but through cross section, you could see that internal tendon that would branch through the muscle itself. The description of bipennate, you notice here, if I complete this figure, there would be a tendon up here, and then the muscle fascicles are running off to make this bipennate shape. And uni, here's the tendon, and just running at an angle just to one side. So these 6 common shapes for skeletal muscle. And we'll finish here now with the description of the terms origins, insertion, and actions. The first 2 truly pertain to the tendons. So what's known as an origin and insertion? These terms are now typically some area on the skeleton. muscles typically connect to the skeleton. And we see that's the point where the tendon precisely will connect. So again, connecting to the various rami and tubercles and tuberosities of the bones, or in some cases, connecting to the skin for facial expressions, connecting to another muscle. And that's what's in common for the terms origin and insertion, just the tendon points where they connect. The second bullet point here, however, this only applies to our appendages. So the muscles that are on our arms or legs. And the reason being this description, the origin tendon, this is always going to be the proximal tendon. And the insertion tendon would be distal. So if we think about our leg, for example, think about your quadriceps muscle. The proximal tendon of the quadriceps muscle, this actually connects to your pelvis, to your coxal bone. That would be the origin, the proximal side. The insertion is distal, and the insertion for your quadriceps is actually all the way down to your patella and then on to your tibia. So the origin for muscles on the arms and legs, that's the proximal, or you could say superior side. And the insertion is the inferior or distal side. This does not apply, though, to the head, neck, and torso. There's no set origin or insertion point for the head, neck, and torso muscles. So again, this second bullet point only applies to the appendages. However, this third bullet point applies to every muscle in our body. So the action is describing the motion, the movement that that muscle now allows. So in all cases, when a muscle is contracting, muscles first can only pull. That's the force they exert, is a pulling force. And they could only pull the insertion tendon closer to the origin tendon. The insertion has the ability to move. The origin tendon is held stationary. So depending on the precise location of the muscle and thus its origin and insertion, each muscle will have its particular action, and we describe those terms as flexion, extension, elevation, depression, whatnot. And again, the structure, the anatomy, the location of the muscle, that's of course what dictates its physiology, its motion. And here's just a nice little sculpture, another example to think of. Here we're seeing the biceps brachii muscle, the origin of the biceps brachii. This is now on the scapula. It's actually these 2 points, hence the name bicep, these 2 branches, these 2 areas here on the scapula. These are the 2 origins. You notice this is the proximal side or the superior side. And now the insertion, you see this right here on the radial tuberosity. This is the insertion. So this is the distal or the inferior point. The action is always to pull the insertion towards the origin. And in this case, the major function of the biceps brachii is to bend the elbow, or the term is flex the elbow. And here's just another figure showing that. So pulling the insertion tendon, applying that force to pull the insertion tendon and physically pull it closer to the origin point.